So when we're working with matrix math, if we have a big letter and it's bolded, it means that it's a matrix. And that means that it can have rows and columns and more than one column. If it is a little letter and it's bolded, that means that it can have multiple rows, but it always has one column. So it's a column vector is what that's called. So these things will always look like this. There are multiple rows, but just one column. So U is little, so you know that U is going to have multiple rows and one column. But the B and the Z and the D, these things, they could have multiple columns. So they can have a rectangular shape. So this is a rectangular shape. That's 3 rows, 2 columns. When we take this and we write it out in matrix form, we will be writing things that look like this. And you can kind of, I guess when you look at this, you kind of think of it as rows. So we can just look at the top and we can kind of see that we have an x equation, one x equation, we have a second x equation. So we have 2 of these processes, 2 of these random walks. And this is b times x. So we've got x1 is b times x1 at time t minus 1 plus an error. So that's a pretty simple equation. And then our x2 is the same. It's x2 at time t equals b at x2 at time t minus 1 plus some error. But we are writing it as this matrix. And then we can write it in this shortened form. And when we do the fitting and the algorithms and everything, it's important that it's written as a matrix. This here tells us the variance covariance, and it tells us how these things covary. Do they tend to both go up together, down together, or are they uncorrelated, for example? For our y's, this would say that there are 3 different observations. They are observing the x's, but we are saying, if we kind of do the math in our head and do that rotation here, it's saying that y1, this observation, is a sum of x1 and x2 plus some error. But this observation is just observing this one. You can see they kind of rotate. This second column goes to the bottom one. So this one observes that one, and this one observes that one. So we're able to model these rich types of relationships between our observations and our data. Okay, so that's a general class. Yeah.
Can you say really briefly what each of the variables of y mean again?
Yeah, so this one is it's what the weighting is for these x's onto the y's. In this case, the weighting is simply 1s. And today, there will only be 1s in this matrix. And it becomes a perturbation or an assignment matrix. It's saying that this y is a sum of, if you do that, it's a sum of these 2. And then in this case, it's assigning this y, by having a 0 here and a 1 here, we allow that, or we set that this is associated with that one. What we're going to be doing today, all the row sums will equal 1, so we won't have any cases like 1, 1 here. They'll always, the row, adding together the rows will always add to 1. And it is, that is what's called a perturbation matrix. And it is assigning which of these Ys is associated with which of these X's, and where it's going to be 1-to-1. So each Y will be associated with one X today. Later, we'll get more complicated. Yes, it is. So that Z will be your loading matrix. In that case, your loading matrix will not be a perturbation matrix, it will not be zeros and ones. It will have values here.
And then so, I guess, how do you, here do you set the weights and then the loading matrix, I guess, how do they differ? I've only come across loading matrices.
Yeah.
Actually.
So when you're estimating loading matrix, your objective is to estimate that loading. You want to know what the loadings are. In this context, we're not estimating this. This is a statement of the structure of our observation. So we're just stating that Y2 is observing X2.
Are there cases where you would assign the loading? I guess, are there cases where they do?
Well, sure. I mean, you don't have to make it zeros and one. You could, depending on what you're doing, you might be, setting up a case where you want Y1 to be 0.8 of X1 and 0.7 of X2, something like that. So you could set that. When we get into dynamic linear models, these are going to be our the weightings in the linear regression. So like if you had, I have an observation and it's equal to some beta times temperature. So that beta is that term, that weighting term. And like in a linear regression, that's just a constant. It has one value. But in a dynamic linear model, that little beta has a T on it. And you allow it to evolve over time. And in that case, these things are going to be those betas. And then what comes in over here are your variables. So like if you had temperature, this would now, this has no time on it, but then you would put a little T on there and then your temperature readings appear there. That's next week. Are these? Yeah, so this is how we're going to incorporate covariance. So things that are driving it. So we could, what I just described with temperature here, that's very specific for dynamic linear models. If we wanted to instead say, hey, I have a variable and I want to try to explain some of this error, this variation by temperature or nitrogen or whatever, then you put that here and you're trying to explain some of this error by your covariate. We'll talk about that on Tuesday. And you can have your covariate either affecting the process. So like, say the process population growth, it could affect population growth. Or you can ask, hey, maybe that variate is affecting my observations. Maybe when it's really cold, all the critters hide, and so I don't see them. So it's not that there are fewer of them, it's just that they're hiding because it's cold. So you could test something like that. Oops. Hey, come here. All right. So we're going to work through an example using a simplified version of chapter 7 in the Mars user guide. So if we go over to the references, if you look here, you'll see this is the user guide for the Mars package, and it has lots and lots of chapters on examples. And this is example, it's actually, I was pausing there because it's example 8 here, combining multi-site data to estimate regional trends. We're going to go through a simplified version of that and kind of walk through the logic. All right, so let's imagine that this blue here is a population. So this population is distributed over this area. And I want to get an estimate of this population. And so I'm going to go out to 3 locations. In one location, I'm going to be doing a mark recapture. In 2 locations, actually, I'm going to be doing 2 separate mark recapture studies. And then in a 3rd location, I'm going to be doing a line transect. And I want to use all this data to try to estimate the total population in this blue area. So I don't think of these locations as like populations. This is one area where the critters are moving around. I'm just taking samples in different, or I'm doing estimates of population size in different areas. So, looking at this, so first thing, my population estimates or my counts or abundance counts in these locations, they are not the total population. They are related to it by some scaling factor, which I don't know. But I know that they are not, in fact, the whole population. So how do we mathematically write out that idea? So I'm going to write out that I think of the population in that blue area as a random walk with drift. That is a exponential population growth in log space. Now, in Ecology 101, when you were taught like exponential growth, you probably imagine something that goes like that. In a random walk context, it doesn't ever do that. These are random walks. They don't like lockstep go up or down. They will kind of wander. And this is saying, it has a tendency to go up, but being a random walk, it can definitely go down also. This is a very common population growth model, especially when we're working in a situation where we don't have reason to think that there's like this tight, what's called mean reversion. So where's that model here, right? If you take a look at these, look at those, they're not like fluctuating like that. Next week, you'll see some phytoplankton data, and that is definitely doing. So we wouldn't use like a pure random walk in that case. But for these kind of data, and this is really typical of vertebrate data, these random walk models tend to work great. Okay, so that's going to be our model. So we have some underlying population trend, which we're going to model with this random walk. And the data are log transformed. And when you log transform a exponential growth model, a stochastic exponential growth, it turns into this additive version. It looks like this. So log abundance at time t is log abundance at t-1 plus this drift term plus errors. All right, so that's this underlying thing we're observing. And we imagine that these 3 locations are observations of this one thing. And we're going to make them pretty simple observations. So each one is just observing this. And the key thing is we have these scaling terms. So remember I said we know they're not a complete survey. It's some fraction of the total. So we don't know what this is, but we know that this exists. We know that we're observing some fraction of that. And this mean here is going to account for that. We're in log space. So a value like, 0.1 here would be, you know, something roughly like 10, that we're saying that this observation is 10% of the total, roughly. And then we also have noise. So these are not perfect measurements of X. They have noise associated with them. We don't know what that is. So here is that observation part in matrix form. Written in a matrix. These are our 3 Y's. Our X's look like this. We just have one X. So we're going to have one column here. We have 3 Y's, so we'll have 3 rows. These are those A terms. They appear as the U. No, sorry, not the U. Sorry, they are the A. Okay, these are our A terms, and then this is our error. We have to fix one of these to 0. Otherwise, it's an indeterminate system. And so what you do is you will treat one, you just sort of pick, it doesn't matter which one you pick. One of these, you will say, okay, I'm going to have the X basically go through this one, and then all these other ones are going to be scaled relative to the X that goes through this. And it's not that you're saying that the population abundance matches to. You're saying, I with this information, I can tell you things about the trends, but I can't tell you things about the absolute abundance. So I'm getting at the structure of the covariance and I'm getting at the trends, but I'm not telling you what the absolute abundance is. You would have to do some other kinds of studies to get an estimate of absolute abundance and then estimate, say, how this one relates to that absolute abundance. All right, so this is the model we're actually going to fit. It's going to look like that. All right, so now we have this. So that seems pretty easy. We're going to estimate this and this. Okay, great. And what about this? We're going to have to say what we think about that noise. We can test different structures, or we can impose an assumption. This is what those errors look like. They're multivariate normal. And we have some variance covariance matrix. And the structure of that matrix says something about how we think the measurements are related. If the variance covariance matrix is diagonal, we're saying these observations are uncorrelated with each other. The fact that our estimate in location 1 is especially high, the error is high. If the error in that is high, it doesn't mean that the error in location 2 would be high. So they're independent of each other. Okay, so even though they're independent, we can still have some differences between them. In this case, we say, well, they're independent and they have different variances. So they're really totally different. In this case, we're saying, hey, they're independent, but there is some information sharing because we're saying they all have the same variance. And in this case, we can use the information from all locations to help us estimate that observation error. This is a great situation. This is kind of what you want more information to estimate this thing. Another kind of common one we might test is something that looks like this. So we feel comfortable making the assumption that the variances are the same, but we think that they covary. So why would you think the variances are the same? Well, let's say you have, pretty similar methods of estimation. It's not like they're totally different types of estimates. However, you think that citability is affected by the environment. So like this example, where if it's really cold, the animals are hiding. So that might be a situation where you think, oh, my errors would be correlated because like they're all affected by this cold temperature. So you could test something like that. This would be an example where it's completely unconstrained. we can have any types of covariance between the observations, and we can have any variances. So all these numbers are different, and all of these, the upper triangle numbers are all different. You don't want that. If you really think that's the case, you need to go back to the drawing board and figure out how to change your sampling or not use all your data. That's just a really hard case. You know, we spend a lot of time when we design experiments so that our study areas are independent of each other. And, you know, hopefully when you were setting up your sampling, you were kind of thinking about that and, you know, maybe put them far apart so that they're not quite so correlated together. All right. So here's some examples of, I just generated with a multivariate normal, and then I use different matrices for it. This is the function in R, in VR norms, what I used, and sampling 10 samples. So this is a case where they all have the same variance, but they are not correlated. They kind of have different sizes. Oh wait, no, this is unequal, sorry. So they're uncorrelated, but the variances are different. And you can kind of see that this one has low variance and this one has really high variance. And they're not tending to go up and down at the same time. So you see this one's down and that one's up. This one, they are also diagonal, so they're independent, but the variances are equal. So it's what we would call diagonal and equal, so equal variance. When you start using the Mars package, these are the shortcut terms for these different types of matrices: diagonal and unequal, diagonal, equal. This one's equal var, covar, so that's all the variances are equal, all the covariances are equal. In this case, I picked a pretty high covariance, and you can see that they all kind of do the same thing. There's a little bit of variation, but they're really highly correlated. And this is an example where it's just unconstrained. And looks like I didn't put too much correlation in there. All right, so let's go through a simplified example of the case study in chapter 8. These are real data from the Puget Sound. This is tracking harbor seals after they were recovering once the Marine Mammal Protection Act was put into place. So it was put into place in the late 70s. And after that, across the West Coast, you know, this region, marine mammals were recovering after being harvested to very low levels. This is what the survey looks like. All these dots are haul outs where they would go and measure them. So here's an example of the haul out. And so they would go, some of them they could survey from land, and then others they had to survey from the boats. So go out there on a particular time and they'd count the number of harbor seals that they could see. They're doing that for all these different sites. And then they divided them into these regions here that are marked here. These are based on studies of the biology, knowledge about the movement of the harbor seals, information about the diet, this kind of information. So the biologists are dividing them into these management units. I think these are actually survey units. The management units are, we'll see in a moment. Okay. No, I take that back. No, these are management units. Okay. So let's say we have that data, and what we would like to understand or make inferences about is the structure of the population of harbor seals in the Puget Sound. And now we're putting on our biologist hat, and we're talking with the biologist saying, you know, okay, what are some of the hypotheses about the population structure that you would be interested in testing? And so let's say they wanted to test this hypothesis. This is a hypothesis about how the population is structured. So we hypothesize that there are 3 subpopulations. There's a south, there's a north around the San Juan Islands, and then there's this population that's out in the Strait of Juan de Fuca, and they're really influenced by the ocean environment. You can imagine these harbor seals are eating something probably fairly different than these, and these harbor seals are probably gone out to the coast, or they're definitely being more exposed to different fish species. So that's our hypothesis about this underlying population structure that, again, we're hypothesizing. We don't know that it's actually structured this way. So we want to be able to express that mathematically so we can test it. And so here is our mathematical representation of that. The x's are the process. So in this case, it is that log abundance for these hypothetical populations, subpopulations. And we're going to say that each of these populations is a random walk with drift. So they may have a tendency to be going up or down. And they each have their own process errors. They each vary differently. So this is saying that, you know, how much they change from year to year, how much they vary is different. So there it is now in this condensed form. There's our U. That's how much they're tending to go up or down over this period as they are recovering. And this is that process variance. And what we care about, what we're trying to get at, is what is the structure of that Q and what is the structure of that U? Were they all recovering at similar rates? And do they tend to vary together, or are they independent of each other? So now when we think about that U, we want to think about different hypotheses about how the population is structured. So we could say one hypothesis is that it's unconstrained. They are each have their own trends. They're doing their own things. Second hypothesis is, no, it's all the same. They all, like, we may see a bit of a different trend, but that just happened by chance, because that can happen. But really, they're, you know, whatever's driving their populations is pretty similar, so we can treat them as all having the same trend, either going up or down. And then a third hypothesis might be, well, you know, everything in the Sound, we think they should have been having the same trend because they're all in this interior waters. But we think that Strait of Juan de Fuca, that one we think was really doing something different than the others. So we should treat that separately. So these are 3 hypotheses we could test. We can do, we do the same for the variance covariance. So let's start here with this simplest one. So what this says is that these 3 populations are independent of each other, but the year-to-year population changes. The variance of that is the same. So they're kind of doing their own thing because they're independent, but how much they vary, like the percent they tend to go up each year or down, is similar. That variance is the same. This one would be, hey, they're all different, they're independent of each other, and they vary in their own ways. So the South maybe has less variability, and maybe the Strait of Juan de Fuca has really high variability. So that's a second hypothesis. Both of these are independent, but in this case, the variability is the level of that is the same, and in this one it's different. Now let's go to this one. So this one we're allowing correlation, but we're imposing some constraints on that. We're saying, okay, so they all they are correlated, but the variance is the same and the correlation is the same. So we're allowing some correlation, but we're only estimating two variance parameters, which is going to be really helpful because it's hard to estimate these. And then this is the unconstrained case. We can have any kind of covariance and any level of variance. This is, that's going to be hard to estimate. We're going to need a lot of data if we're going to try to do that. And how this will turn out when you're doing model selection, which is how we're going to compare these hypotheses. What's going to happen is that model selection, it might say, hey, that fits the data really great, but it will say, no, there's no support for this because you are estimating 1, 2, 3, 4, 5, 6 parameters. It's a lot. All right, okay. Okay, so there we've got the mathematical structure. Okay, so great. So we've been at the whiteboard. We are standing up there at the whiteboard with the biologist coming up with this hypothesis that are these hypotheses that we want to test. So we have 3 hypotheses about the trends, and we have 4 hypotheses about the variance covariance structure. So those are, 3 times 4, 12 different models that we're going to test about our underlying population structure. Okay, so now got that done. The next thing we have to think about is how do our observations relate to those subpopulations? So I realize this looks kind of like, every time I look at this, I think, oh, those are seabirds. They're not seabirds, they're harbor seals. I'm just kind of looking at the boat. Okay, so we have for these regions, we actually have 5 different sampling locations that are overlapping. Or here, let me go back here. So this is what that actual reporting is. So we don't have these points here. We have reporting for these regions. So we're going to call these 2 south. We're going to call 5 and 4 the north, and 3 Juan de Fuca. So that's where this is coming from. There we go. And so now we need to specify mathematically that structure. So we want to say, okay, this one's pretty easy. This is just, this one is measuring that one. For the north, we need to have that San Juans and Eastern Bays are associated with the north population, and then we want these 2 associated with the south population. So this is where we get that Z matrix going. So this is going to be that perturbation matrix. It's going to tell us how observations line up to our hypothesis about the population structure. So this is this hidden population structure. So we have, this is our 3, I've color coded them. You can see we have a 1 in the Juan de Fuca line, and that's saying that this one is associated with that. We have 1 location associated with this underlying population trend. This one, we have 2 locations. This one and this one are associated with the north, and then for the south, we have these 2. There's no reason that we had to have it ordered so that these 2 are together and these 2 are together. I did it that way just so it'd be a little less cluttered looking. We could have, you know, reorder this so like that 5 was on top, say. This matrix is going to tell us how these things are lining up to these things. Okay, so we have that, and now we're going to have these observation biases, and now we have these measurement errors. Okay. Like with the process, we also have this observation variance covariance matrix, and we need to make some assumptions about this. We can't have this. Certainly hope that the observation errors don't look like that. These are the typical ones that you would assume. So you want to hopefully you can convince yourself that this is a reasonable assumption that the observation errors are uncorrelated. In this case, we are taking averages of lots of different sampling locations. And these sampling locations are separated. Yeah.
The question related to your slide, can I have like an observation time series led to more than one process? So it would be the other way around.
Yeah, you could. So when you are doing, say, dynamic factor analysis, and this Z is a loading, this Y will be a loading, it'll have multiple values in a row. And so it will be an observation of loadings of these Ys. Yeah. But there could also be other reasons why you might think that. So maybe maybe one of your sampling regions is kind of a mix of these. And so you think it's a combination of them. You could do something like that. All right. So this is our final model. Yeah. Yeah.
Do you in practice find cases where like you want to specify an error variance matrix where like between diagonal equal and diagonal and unequal, where like I'm thinking if you had like, for example, similar survey methods for some observations and a different survey method for another, where you have like 2 or 3 different variance terms for multiple sites.
Right. So what you do there, and that's a really good question. So you can say, so you see these numbers 1, 2, 3, 4, 5. Well, I could say that these 1 and 1 are same variances, and these are all different. Yeah, I could do something
That's
like that.
in practice that comes up. So okay, not infrequently.
Yeah, that comes up not infrequently. Yeah, like especially if like you're combining, say, types of surveys that have really different errors. It also comes up when let's say you're trying to get the temperature of a lake, and you have really different types of sensors, and you know that some sensors have really high variance. So you would model that by saying they're different. There are situations where you might say, hey, I know what the variance is because it's a temperature sensor. I know what that is. That does not come up in practice very often. The reason is that sensor variance is, for us, typically not the only variance out there. Not the only reason that temperature is varying. Right, so it may be that you're out in the lake, and as the sun goes, as the clouds come and go, that you have like sensor variability, but you also have variability because the clouds are coming and going. And that's adding this extra source. And in one part of the lake, you also have these trees that are like it's all kinds of a million things beyond your sensor variance. Okay, so here's it in condensed form. This is the identity matrix. So it is a what do I do with it? I didn't write all the zeros, there's 3 of them here. So we have ones along the diagonal, zeros everywhere else. It's called identity. If we multiply that times another matrix, we just get, like, if we multiply identity times X, we just get X out. Okay, and then here are our U's. And then, yeah, our errors. So those are the things that we have. What are we estimating? We're going to be estimating the U's. We're going to estimate the A's, but these are the scaling parameters. We're not going to care about those because we're trying to get at whether the trends are different. We care about this. And then we care a lot about that because we're asking things about population structure. And we might care about that. You know, if we were doing something like saying, hey, are the ship-based estimates, are they more variable than the land-based ones? Do something like that. So that was all the steps we go through to set up the things we're going to test for a hypothesis that there are 3 underlying subpopulations that are potentially covariant. But, you know, in talking to the biologists, they would like to test some other hypotheses about how the population is structured. Because these have implications about how they manage the harbor seals. So they would definitely like to test this hypothesis. So this is the hypothesis that, you know, the harbor seals are moving around enough that we might as well treat it as one panmictic population. And we can model that by saying, okay, so effectively what we're saying is that these are all observations of one random walk. So that look like, we have those, you know, these are Y's, these are our, those are our 5 locations. This is our Z. The Z, and we just have one X. Each of these is observing this X. They look different because, one, we can have different scaling. They can be moved up and down relative to it. And also, they can have different variances. So some of them could have really high variability and others have really low variability. So we can test whether that hypothesis or that structure is consistent with our data. The biologists might want to test this. They say, well, you know, we think that the San Juans and the Strait of Juan de Fuca, that, you know, based on what we're observing, we think, you know, these move a lot around. There's a lot of mixing. Or actually, okay, step back. They go out and they measure things and they say, okay, the San Juan group, they kind of stick together. They're doing their own thing. They don't really leave these little bays. We think that's can be treated as one population. The San Juan group definitely, they're doing their own thing. We track them, they're going out to the ocean, so we want to treat that as a subpopulation. But this inland, we think this whole thing, we don't think it's separated into north and south, because we are, we've been tracking them, and they just kind of go up and down here. So what we want to test is the population structure actually like this, and then two separate ones here. So in this case, we still have three underlying populations, but these three are observing this one. This one is observing that, and that Y is observing this one. So still three X's, but different structure to how the observations are related to the underlying behavior. And you can imagine, all kinds of different structures you could test.
Yeah, I guess you would run all of these.
Yeah, I'm going to show you an example, a real world example, right now. So this was a study that Eric and I did. It's now been many years. We were approached by some American and Mexican biologists who study these sea lions in the Gulf of California. So they have these study sites all up and down here. And they, people for a long time had hypothesized about what governs the population structure in this region. And they asked us to help them analyze the data and see if they could test the support for different hypotheses. And so we had a variety of sessions with them. We up against the whiteboard, and we are talking with them. Okay, what do you think causes the fluctuations in the population, and why do you think they'd be correlated in one way versus another? And they had four hypotheses. One was diet is what causes correlation. The other one was disease, and then DNA, some movement of breeding individuals, and then just distance. So like, it's just like ones that are farther apart are more different. And then two models, two null models about like, this one is fully structured, means that each of them is its own subpopulation. They didn't think that, but that was like one of the null models. And then at the other extreme, we can just treat this as one big pan-mythic population of sea lions. So they wanted to test those. And let's make this a little bigger here. So we we're testing structures of the U's, the Q's, and the R. This is the sampling errors. This is the population covariance, and this is the trends, if they're the same or different. And these are shortcuts to the different structures that we were testing. These are all like diagonal ones, so they are independent, and then this is our correlated ones. And we so we had those structures, and then we had different types of circles here that would correspond to the different hypotheses. So I forget which hypothesis this is. It looks to me like it might be the distance hypothesis. So let's say this is the distance one. Well, then for the disease one, they had samples of where disease outbreaks were happening. And, they had a different clustering. And then they also had information about how the animals were moving between the rookeries. So they had a different collection for like their DNA hypothesis. So these hypotheses are basically different Z matrices. And we fit them all and used, this table is AIC, yeah. Yeah, this one is using AIC. And you can see this was the model that fell out as having the lowest, and actually a lot of them they're much, much, much higher. They just didn't have any support. A lot of these are really big. Generally, if the AIC is within about 2, 3, then you say, well, you can't really distinguish them. And you can see that I think we had any that are within 3 of that. Yeah, this came out as a very strong contender. And what this was saying is, first off, that it was simply distance, but the populations were correlated together. Makes sense, they're in this similar environment. And this is interesting, the observation errors were not unique. And the trends, however, were unique, meaning these populations here were tending to either go up or down differently. Again, maybe not too surprising, probably the ones near the down here eating something different. All right. So lots more examples of things like this. And first off, we have the lab book. The lab book is something we have put together over the years. Each time we teach this, we'll add another chapter of an example showing an example of how to do something. So more examples there. We also have a number of these population analyses in that. You can find links to this on the resources on the webpage. We're about to take a break. Just about done here. Okay, so that was an example where we're asking questions about underlying population structure. What are some other really common things that people use? Basically, exactly the kind of model that I've just shown you. What are other things they do with that? So another common thing would be, hey, I've got all these sensors. These are actual weather stations in the Methil Valley in Washington. I would like to take these and combine them with a regional temperature estimate. So I can combine lots of station data into a single metric. I can take into account that the variance of these different stations can be different. Again, some of them are out in the field, some of them are like under shade, totally reasonable. They would vary in different ways, but they're all somehow related to this overall temperature in this region. So we can use these models to do things like that. We had like lots of individuals that we're tracking, and we have their growth rates. And what we want to get is a population average of these growth rates. So people would use this to do those kind of things. Here's another really good example. This is actually from the lab book. So imagine, this is SNOTEL measurements. So it's either snow depth or it's SWE. It's water equivalent within the snowpack. And we have these measurements up here in the mountain. Now imagine that you were doing a study where you needed estimates of the snow, of the SWE at every year, because you're using this as a covariate in some studies that you're doing. Well, the data are the points here. And you can see we have a problem because some of our regions don't have a lot of data. They're missing data. And so now you're stuck because you really needed those for this next step in your analysis. And instead of just throwing up your hand saying, I give up, you can take this class of model and use the information in the data, because it turns out that the snow in different regions is not uncorrelated with each other. I mean, it makes total sense, right? That it's not uncorrelated. And these regions that are close to each other are pretty correlated, and they're more correlated than the ones over here. And we can estimate that structure using the models you've just seen. And we can use that to make estimates of what the SWE is for these other years. And now you have an intelligent estimate of the missing data, rather than doing something like, I'll just say that it's the mean or something like that. You're using all the information about how things were varying in the sites where you do have data. So this is a really common way that this class of model is used. This, like I said, this example is in the lab manual. All right, so let's stop there. Let's take a 5-minute break, and then we are going to finish up by opening up our studio and running through some code, starting to fit these models. Okay, I need to. All right. So let's finish up and get to those scripts. So we're going to be using the Mars package, and I want to briefly show you what the syntax looks like. The function is Mars. You give it data, and then you give it model as a list. And the key thing to know about Mars is it wants matrices basically for everything. It wants a matrix for your data, and it wants a list of matrices here. And it is 1-to-1 with the equations that you've been seeing. So I showed you that the Y has each row are the observations. So like with the harbor seals, we had 5. So that means that your Y needs to be 5 rows, and then the columns will be the time steps. So if you had 20 years, it's going to be 5 rows, 20 years. It needs to be a matrix. Let's see what that looks like here. Not that example. Here's a better example. Okay. So let's say we have a model that looks like this and looks like that. So first step we always do is we write it out in matrix form, just like we would on a piece of paper. And we translate that into a list for each of the parameters. They are named B, U, Q, Z, A, and R, and they are capitalized like that. So let's start here. So that's B. What is that? That is a 1-by-1 matrix with 1 in it. So if it's a 1-by-1 matrix, you don't have to say 1, 1, 1. You can just say 1, but it is a matrix. So B is a matrix of 1. That may seem silly, like, why don't you just say it's 1? But it wants a matrix. It wants you to be absolutely clear to it, for it. U So this is a matrix, a 1x1 matrix with a U. In Mars, if you put things in quotes, it is something to be estimated. It doesn't matter what you call that thing. If you put 1, you were to put this in quotes, Mars would say, oh, you have a parameter that you would like me to estimate, and you have named it 1. I will do that for you. So if you don't want it to do that, if it is really a 1, or more likely really a 0, then you need to make sure it's a number and it doesn't have any quotes around it. So you kind of walk through and you make this list, you give it names. This is what U is, this is what B is, this is what Q is. In this case, I fixed Q to be a 0.1, so it's a fixed value. I can do that. I don't have to estimate it. So here I'm going to fix it. What about these cases? So this one is my Z is a 2-row, 1-column matrix with 1s in it. So I have 1s, 2 rows, and a 1. We kind of walk through all of these and fill it out. Fortunately, a lot of these have defaults that we're not going to have to make this thing every time. Okay, so let's jump in and start playing around with it. I think that's all you need to know so far. It's just that the model list is matrices and it's 1-to-1 with what you write on the blackboard. Okay, so I want to stop sharing and I want to share something else. Okay. So what you need to do is go. So if we go to computer labs, and if we go to the repo folder, it's going to take you here. But we don't want lab 2 because we're not doing that yet. We're going to go lectures week 3 right there. And there's a bunch of scripts in here. If you have cloned this, navigate to lectures 3. If you have not done that and you don't want to hassle with it, that's fine. You can click on this and do download the raw file. Whatever works for you. We're going to start off with Mars example 0. Where am I? I come back here. Here, share. There we go. Okay. Good. Wow. I would like to make you bigger. No. Let's do, let's say 0. Yeah, I'll do 0. I'm going to do the one with comments. Let's do view, zoom in a little bit. There we go. If you have installed the ATSA library, you already have Mars. If you don't, you can go into packages over here and install it. So go through this, we're gonna set that up. And we are going to start with looking at the Harbor Seal data that comes in the Mars package. And let's go ahead and run that. and take a look at what that data looks like. Okay. Yep, there we go. So you can see that it is a matrix. That might not be obvious, but I can say class dat, and you'll see it is a matrix. So it's a matrix, and it has looks like 4 dat. I think it's 4. Yeah, 4. So it has 4. I was just making sure it wasn't cutting anything off there. So we have 4 places that we are surveying, and we have 22 years. You see that there are missing values in there. That's totally fine. These models can handle missing values. It's going to use the information from the other time series to help it fill in estimates for that. And so we're not going to do plotting the data. We are going to first fit a model with one underlying state. So one population process. All sites have a different observation variance. So they're all observing this one state. So, okay. So I've got 4 of them. One is trying to figure out what the map looks like. Z, this is my Z. 4, 1 column, I've got 1x. And it is going to be plus an A. It's going to look like this. You generally will not have to care about A. So you're not going to have to pass an A. The Mars model is going to do the right thing for you. I'll just show you what it looks like, but we're not going to be generally adding that. I don't know if I have that in the script, but as I might have it in there just kind of for pedagogy, but in general, we don't have to specify this. Mars will do the right thing. So that's our A, and then we have our error, and that BT is going to have some multivariate normal, and it's going to have a 0. This will always be 0. The thing is, the R is what we have to specify, and that is going to be a 4 by 4, and we are going to either specify that it is diagonal with a value here, or some kind of variance covariance structure. So it looks like that. Our x's are simple. We said O's just has 1, so that's easy. X t minus 1. So this b equals 1. U, we're going to estimate that. And then we have WT. WT is I sketched out what it looks like mathematically, and now we can fit it. So here's our model list up there. Yeah. Yes, and I did write everything out for pedagogy purposes. So the B, what does it look like? It's a matrix 1. What does the U look like? We're going to be estimating that. Remember, it has to be a matrix, so I wrap it in matrix. It's a matrix. It's a U. I put it in quotes because I want to estimate it. Q, that variance covariance matrix, is also 1 by 1. I'm going to estimate that Q, so I put it in Q. You can call the things in quotes whatever you want to call them. The Z is a matrix with 1s. It's got 4 rows and 1 column. A, that's the default, is scaling. In later cases, I won't be putting that there because that's just the default. That's the intelligent thing to do. Just let it do that. And then R, instead of writing that whole R, 4x4 matrix out, I'm going to use a shortcut. So this is one of the shortcuts, diagonal and unequal. Let's see. XO T and at X has to do with the initial conditions. I put those up there for completeness. You will just ignore those. Mars will do the right thing for you. It will take care of properly estimating those for you. And generally speaking, you won't be caring about what those are. It's just something that to fully specify the model has to be there. So we make it a list. So there we go. If we do, mod list 0, you can see it's a list. Hopefully. If you ever want to know what something is, wrap it in class, it'll tell you. To fit something, whoa! In the Mars package, the function is Mars, you just give it the data. Remember, it has to be a matrix, and then you give it that model list. Let's go ahead and do that. So we fit it. Boom, that was fast. So it fit it, and it gave us our estimates. These are the scaling parameters. I'm not going to care about those. And these are the different R variances. You can see the Strait of Juan de Fuca and the San Juan Islands are kind of similar. And then the inland region is a little bit less variable. That's kind of interesting. And then here's our U, the trend increasing. As I said, these are data as the population is recovering once the Marine Mammal Protection Act is put in. This is our process variance. It's really small. This is pretty typical for vertebrates, that it's going to be something small like that. All right, so we got that, and let's do something fun with it. Let's plot it. Auto plot. This is going to be your basic function. It's going to give you all your diagnostic plots. Let's look at it. So it's going to go through a series of plots. Here's the first one. Let's get this up here. Okay, here you go. So this is the state. This is our estimate of that single population abundance time series. So this is the best estimate of that. And it on the bottom, it'll tell you what you're looking at. This is an estimate of the X, the state, conditioned on the data from 1 to T. It is conditioned on all the data. So it takes into account all the data, all the information that you have, and gets that estimate. And these confidence intervals are the actually, I don't remember, is this 80% or 90%? Don't remember that off the top of my head. So it's the confidence intervals on the expected value. So this is the expected value conditioned on all the data. And this is the confidence intervals about that expected value. Hey, come here. Let's go to the next one. All right. So look down here. What is this one showing? Get out of there. Okay. So this is the fitted value of Y of the data conditioned on the data, on the observations of the data. So this is the these are the data. This is that we had like the Y equals for this one, it's that Y equals X plus some error. So this is that in this case, it's just the X because that's the expected value of the Y's. why might you want this? Well, you might want this if you wanted to get estimates of, you know, this is your average estimate of what you would have seen if you had collected data. We don't use this one so much. Usually people are using the previous plot. These are the model residuals. You don't want these to look too alarming. What is alarming? That is something that doesn't look kind of fluctuating around the line at all. You might find this a little bit alarming, although it's not that much data. So you wouldn't want a lot of your points to be outside of these gray bars here. So, you know, it says, and it gives you a clue here, you know, these, you know, 95% of your residuals should fall within these points. Why is it collapsing here like this? It doesn't have confidence intervals when there's no data. So there's nothing alarming about it looking like that. It's just that there was no data. Yeah, this one's looking for temporal trends. You can see the San Juan de Fuca, not so great. So what is that telling you? That is telling you like, this model where I only have one X and I'm saying that all these regions are doing the same thing and they're tracking the same underlying population. Maybe not such a great model, certainly not for Strait of Juan de Fuca. Maybe not for Eastern Bays also. So that's calling into question your assumptions about that underlying structure. Okay, so these are our normality test. These are QQ plots, and these ones are one of those things where someone says, is it close enough? And they're like, yeah, it looks cool. Yeah, it looks close enough. And yes, these all look pretty normal for that much data. When things don't look normal, typically you take one look and you're like, whoa, that's not normal. Like it looks like really weird. Like a really non-normal would be something that looks like this, like that. And you will see things like that. That's when you should be concerned. These are your state residuals. This right, you like got some outliers going on. My guess is that's that Strait of Juan de Fuca issue. This is testing your autocorrelation. You shouldn't see anything alarming here. So these should be uncorrelated. There's nothing really alarming there. All right. Good. So we've gotten through all of those. There are a few different ways that you can standardize residuals, and yes, it is using the Cholesky of the variance covariance matrix to standardize them.
Is there anything to do with computation? Is that like
No. It's a very standardization for the residuals. All right, so let's go. We'll do one more before we break and go into the lab here. Okay, so let's do a different one. Let's do here a model with 4 underlying states. So each, so instead of having one X there, we're gonna have 4. So let's see, what does that look like? We're gonna have 4 X's, 2, X1, X2, X3, X4. And that means that this has to be 4 and 4. And this will be diagonal. What that does is makes this Y observing that X, that Y observing that one, this one observing that. So if we have 4 of these, so it's going to be diagonal there. And then our A is going to be 0, actually, or has no information to estimate the scaling when it's 1-to-1 like this. You don't have to worry about this. Mars will do the right thing. And then we will have our variance. And so now that Q is going to be this 4x4 thing. We can ask some questions about the structure of that.
And you were saying this is testing that there's 4 separate underlying?
Yeah. 4 separate, so they're each doing their own thing. This one. Okay, here's how we change it. So our B is a diagonal with 1 and 4 rows. Just as a note, the default is for B to be a identity matrix like this. Technically, I could have left that out and it will make that a diagonal. Our U matrix. So this is the observation, and this is R. It is a 4x4, and then our X is where that Q and U are going to come in. So our X, we've got 4 of them. And then I'm going to leave the B out because we're just treating this as a random walk. So this is a random walk with no interactions. 1, X2, X3, X4. We are not going to be dealing with the B until way later in the course. So we're always just going to have an identity matrix here, which is that diagonal with the 1s. Okay, that's that. And then our U's. 4 there. And then we have the W's are going to have that multivariate normal 0Q. Q is a 4x4. That is specifying how these are covariant together. So do these 4 populations, do they tend to go up and down together? Do I have good and bad years together? Or are they independent? And whether one has a good year, like it doesn't give you any information about if the other has a good year. Okay, so here's our model list. We got our B. It's just an identity. And our U matrix. What are we specifying here? When I wrote it on the board, I had a different U for each population, but here I'm going to say it's one U. So I am saying, okay, I'm going to test a model where they're all separate, but they have the same trend. What about my Q? What am I testing here? Diagonal, so they're independent, but equal variance. So they're independent, but they have the same year-to-year fluctuations. Z matrix, it's going to be, you see, that's diagonal there. Scaling, that's just the default. We don't need to care about that. R is our assumption about the observation errors. Here I'm saying it's diagonal. They're independent. Unequal means they have different variances. Let's go ahead and fit that. So they fitted. Got a bunch of estimates. Let's check out the plots. You can do either plot or auto plot. I like auto plot. I think it's prettier. One. Yep. All right. There we go. These are the states. So these are the 4. So they're a little bit different, right? We said they're independent. We did force the variance to be the same, but they are, because they're independent, they can be doing their own thing. Why are they all kind of going up the same amount though? Well, I imposed that. I said, hey, I want to have a U that's all the same. So in general, they are going to have the same trend. It's stochastic, so there'll be some variation, but they're going to tend to have the same trend.
You sees within have figured out what the individual trend is for each.
Yeah. Let's do that. Let's do this. Okay, so if you look here, you'll see that U is 0.05. Let's do the shortcut for all different is unequal. And let's run that one. Oh, sorry. Run that. And run that. And now you can see it's estimating these different ones. And so this is Greater Watertuca going up a little faster, San Juan Islands going up even faster, and then the Eastern Bays and Puget Sound going up a little slower. If I plot that, auto plot fit 1. Now, it's a little hard to see because the scales are different on the y-axis, but the these two are going up a little faster than these two. All right. I'm going to stop there. I'm showing that. There's lots more scripts you can play around with. You will also find in the repository. Is it in the repository? No, I think it's on the website. Let's go back to the website. Reference sheet. So if you click on that, it's going to give you some a table with all those shortcuts. So you're going to want to have this handy because you don't want to have to create all those matrices if you don't need to. If there's a shortcut, you want to use the shortcut. One tip I will say is that when you get into creating Q and R matrices, if you if you do end up creating your own, for example, these examples down here are creating your own, and you might want to do that if you have really specific structures. So you want to say, hey, I want these populations to be covariant together. I want these to covary together, and I want everything else to be uncorrelated. So I want it to be 0. Like you can do like really complicated things. If you get into that, always make sure to print out that Q matrix. Is super easy to make mistakes there. All right. So lab this week is very realistic. You're going to be analyzing salmon data, endangered salmon data. There are your teams for this week, and it's in lab 2 in the repo. These are data from the Coordinated Assessments Partnership, and I downloaded the data this morning. That is not Columbia River. That is a mistake there. It is called ESA Salmon. And when you load the data, load it in like that. It has all these ESUs and DPSs, and you'll be looking at different ones. So some of you, so team 1, you're going to be looking at Oregon Coast Coho. Team 2, you'll look at Snake River Spring Summer Chinook. And team 3, you'll be looking at Steelhead. When you look at these ESUs, they're divided up into these major population segments. This is just kind of an example to get you an idea. And you'll be asking questions about whether there's support for this type of population structure. And then on Tuesday, we'll introduce covariates. You'll also ask, hey, is there any support for these populations being affected by the marine climate or other climate variables, whatever I come up with this weekend. I show some code here. This is a function I wrote to make a little plot. And let's look at one of these. So this is group 1. This is the data that you'll be looking at. This is the Oregon Coast Coho. You're going to analyze that, and these are the major population groups. You'll go through that and ask some questions about that. Let's see here. Here is the steelhead. A little bit less data. And then spring, summer, Chinook. You've got a bunch. This is kind of interesting too. You can see that you have some gaps here. Why are we doing that here? Oh, I shouldn't have that. That's all the data by run timing. You don't need that. Okay. So these are the questions you'll be asking, creating estimates of the spawner abundance for all the missing years, getting estimates of the decline, estimating support for the major population groups, and evaluating the correlation with the climate indices. I have a sample analysis here for Upper Columbia steelhead. Walks you through how you can go about that. You'll also have last year's examples. They weren't working with this data, but they're asking similar questions with some different data. So you can also get some ideas from what they were doing. Their repo has the same name, but it's 2023. So just put 2023 there. So feel free to go and look at what the previous students did two years ago to get you some ideas. Actually, I'll show you what that looks like here. Yeah. So if you go there, you're going to see their work. So here's what they were doing. So you can poke around there, maybe get some ideas. All right, I'm gonna stop there, let you guys start jumping in. Make sure that you can get the, you get in here and get this and run that. Make sure you can run that before you leave. You should be able to run it and just say, run the whole thing. If you run into any trouble, I can help you now. But fingers crossed. Yeah.
I noticed in the instructions it said that there was gonna be a discussion about how to find the climate variables. Is that in there?
That'll be for Tuesday. I'll talk about how to include the climate variables. Yeah. I need to grab some climate variables too. I haven't decided what. I have lots of them out there. Okay. Put in. I have a new project I was working on is getting all this modeled stream flow data for all of the West Coast, for all of the rivers. Every river segment. I discovered it. A colleague needed some help, and so I was like, oh wow, that's so cool. So I have that now. So now I've been trying to think of like, how do I, especially for Oregon Coho, that's right next to the ocean. And you get this really steep, like it's down to sea level and then high. So I don't know if it's Coho is really affected by changes in stream flow. I know that for the Olympic Peninsula, Chinook, Steelhead, they have there's been declines in stream flow. So all right, okay, I'm gonna stop talking here. You guys can get in your groups, make sure you can run the stuff.